Hello, I'm Charles Cantor, the Chief Scientific Officer of Sequenome, and I'm going to talk to you today about how we use human genome sequence information to try to improve medical care. Now, I call this subject non-invasive personalized genomics because, as you'll see, we use extensive amounts of human genome sequence information in order to draw medically useful conclusions. To give you an outline of what I'm going to describe today, I'm going to give you some background on the current status of the human genome sequence information and give you a brief introduction to motivate why we do the kind of non-invasive testing that I'll describe. And then I'll show you detailed examples in two areas in non-invasive prenatal testing, which is a mature commercial product, and in non-invasive characterization of cancer, which is still a research tool. So it's 10 years or more since we've had complete human genome sequence information, and it's fair to ask how useful this is. And the answer is complex. It's extremely useful in a few diseases like cancer and in prenatal testing. And it's not useful in most complex diseases. And in a few minutes, I'll show you why that is. Well, the cost of human genome sequencing is dropping substantially every year. And soon, it's probably going to be possible to determine a complete human exome, if not a complete human genome sequence for essentially trivial cost. And we have to ask several questions as we view that future. Is it going to be useful? What's the impact that it's going to have on various aspects of medicine, including diagnostics and therapy? And let's start with, once we have everybody sequenced, is there going to be any future left for DNA diagnostic tests? And I'm going to show you in this talk that there is a very rosy future for DNA diagnostics, but in aspects that may surprise you. So we don't have a $100 genome today, but it's possible to have inexpensive tests that characterize a substantial fraction of one's genome. For example, there's a test available from a company, 23andMe. And so what do you learn if you have a test like this done today? Well, the first person I know that had this test is Mark Chi, who was then in Illumina. And Mark learned that he was Asian. And that's about all he learned. So why is this? Well, will a $100 complete genome or complete exome sequence be more informative than 23andMe? The answer is depends what you're interested in. So if the more detailed sequence information we have about a person, the more accurately we can reconstruct their ancestors. And if the person has a very serious single gene Mendelian disease, the sequence will reveal that. And if they have an unknown disease, it will be tremendously informative. But in most cases, a person who has this serious single gene disease already knows it because most of these diseases have dramatic phenotypes. Our problem is that for all but simple diseases, we're not very good today at predicting disease susceptibility or the particular phenotypes associated with the disease from pure genome sequence. Okay? Now there's an exception. The exception is age-related macular degeneration, which is the largest cause of blindness in old people, because although that initially was viewed as a complex disease, it turns out that only 5 genes really dominate most of this disease. And we know a small number, something like 13, genetic variations that are common in the population that give us a pretty good predictive value of a person's risk of developing the blindness that associates with this disease over time. But this was the exception. If you like, this was the poster child of whole genome analysis. And most other complex diseases are truly complex. And the number of genes involved is simply staggering. And frankly, this is beyond our computational and perhaps even beyond our intellectual ability to disentangle what's going on in the pure genetics of complex disease. And the reason is not only are many genes involved, but the environment involved. The environment is involved as well. Now, most phenotypic traits in humans are roughly half genetic and half environmental. And we know this because we have twins fairly frequently in the human population. And we have monozygotic twins, which are genetically identical. And we have dizygotic twins, which are half genetically identical. And if we look at disease susceptibility in pairs of monozygotic and dizygotic twins, we can calculate a reasonable estimate for heritability. That is the fraction of the phenotype that depends on the genotype. But I'm telling you, in an average situation, it's about 50%. Okay. Now, we don't really have any way to change the genetics today. And so one of my recommendations in this talk is that if you're interested in developing drugs, you're best off to concentrate on environmental risk factors rather than genetic risk factors until we have either more advanced methods of drug discovery or we actually have gene therapy. Well, it is possible to unravel the genetics of complex disease, and this has been done in large studies in a number of major diseases. But the results, as I've already hinted, have been somewhat disappointing. They honestly haven't been that useful. To make these studies better and more useful, we need even larger studies, and it's not clear where the funding for those larger studies, perhaps hundreds of thousands of patients, will come from, because it's not clear there will be commercially useful outcome. And one of the problems here is that disease is really not a good endpoint for most complex disease. We need molecular phenotypes. We need phenotypes closer to the disease mechanism. And in the rare cases where those molecular phenotypes have been used, the picture becomes clearer. But in the end, what we're going to learn, I'm afraid, is that each major disease is tens or hundreds of different diseases. And so in terms of the populations for each, we're going to wind up making major diseases or orphan diseases, and that's not completely encouraging for developing simple therapeutics. Okay, now there are people, I'm sure, who think what a wonderful commercial opportunity it will be when we can sequence everybody inexpensively. But it turns out that that attitude is actually pretty short-sighted. The reason is that you only have to measure the genome sequence once in a lifetime. It never changes. And once we know parental genome sequences, we can predict the genome sequences of their children with great accuracy, because all we have to determine is the meiotic recombination breakpoints, which is a far simpler task than determining a whole genome sequence. And the genome, with rare exception, is mostly the same in every cell. So I honestly don't think that $100 genome sequencing is a long-term, sustainable, commercial opportunity. So what then is the future of DNA sequencing? Well, the ideal diagnostic test has to be repeated often. And fortunately, we have many examples where it's clear it's going to be medically useful to repeatedly sequence portions or even entire genomes during a patient's lifetime. So here's a list. It gets complicated, and I'll go through it quickly. Remember that in mammals, we have 5 DNA bases, not 4, and 5-methyl-C. Well, we have 6 hydroxymethyl-C as well. These modified Cs are very important. They're different in every tissue. They change during the lifetime. They have enormous predictive value for gene activity. And so epigenetic measurements are valuable as a signature of how the genome is interacting with the environment. And in the future, I suspect we're going to want to characterize epigenetics often. The challenge is that it's different in every tissue. And so it's not clear what tissues we can access easily that will represent the clinically important epigenetic variables. Gene expression, in some ways, is a mirror of epigenetics. It has high dynamic range. But honestly, our experience at Sequenome is that trying to measure RNA in clinical samples in a real clinical laboratory is challenging and requires very strict adherence to operating procedures. Metagenomics is the non-human component of DNA, and most of our DNA is not human. It's bacterial or other microorganisms, and that also changes constantly as a result of the environment, and it's very useful, but it's still very expensive to measure. In addition, in some cells, the germline is not constant, so in cancer, somatic mutations occur, as you'll see later, and these determine the properties of tumor. And in the immune system, of course, there are DNA rearrangements. So before I proceed into more detail, let me just remind you that I'll take question and answers during, you could submit questions during the talk electronically, but I'll answer the questions at the end of my presentation. Okay, so what I'm suggesting is that instead of focusing on using human genome sequence information to explore genetic risk, we instead use it to explore coupling between the environment and the genome and focus on signs of disease onset, progression, minimal residual disease, disease recurrence, and also focus on the one thing that turns out to be relatively simple, which is the genetics of drug response. So pharmacogenetics, now sometimes called pharmacogenomics, both in the germline and in somatic tissue, is very informative and basically is revolutionizing much of how we think about cancer, and is simple because in most cases the genetics of how a drug, a very small molecule, interacts with its target or the enzymes that metabolize it. In most cases, those variables are genetically simple. The other area that I think we should focus on is biomarkers, which are characteristic to either disease in a tissue or the disease process. And that latter focus will be what I concentrate on in the remainder of this talk. So biomarkers can be anything that you can measure in a patient, but I prefer DNA for several reasons. It turns out that it's fundamental to the disease process. It's very stable in clinical samples. And we have PCR, which means that every sample and every target more or less can be approached with a common protocol. And this is extremely convenient, and it's just not available for any other kind of biomarker. So at Sequenome, we focus not exclusively, but mostly on DNA. So my goal is to get clinically useful DNA samples from patients without hurting them. And I think the advantages of being able to do non-invasive testing, the advantages are compelling. The most important advantage is you can repeat the test as often as you like, and the patient is not likely to complain. So you can get DNA from a variety of sources in the body, but blood is the most commonly used for a variety of reasons that I think are, at least today, pretty obvious. So, to motivate you to try to think about why blood is such a good source for biomarkers, let me pose what at first might seem to be a very peculiar question. What do Alzheimer's disease, cancer, and pregnancy have in common? In all three cases, we have a chance, I think, of finding DNA biomarkers in the blood. And the reason is because in all these situations, and many other clinical situations, cells die. This primarily happens by apoptosis. And when they die, they release small DNA fragments into the circulation. The released DNA is relatively stable, at least in pregnancy. It has about a 15-minute half-life. And as long as there are differences between the DNA that come from a fetus or an Alzheimer's brain or a tumor, as long as there are differences between those DNAs and the DNAs that normally populate blood, which come from broken white blood cells and endothelial cells, we can characterize those differences and infer properties of a tumor, the brain, or a fetus without touching it. And these can be DNA sequence differences, they can be copy number differences, or they can be epigenetic differences. But we have one problem, and it's the problem that's compromised these attempts to do non-invasive characterization of DNA in the past. The DNA is different in the blood in those clinical settings, but there's not very much of the different DNA. And in fact, there's so little that we run into a basic problem of physics called stochastic noise. So if you have less than a few hundred molecules of analyte, then it turns out that it's impossible to precisely quantify how many molecules you have because of statistical fluctuations. And if you have 10 molecules or less, you may miss a rare sequence or an otherwise informative DNA fragment altogether, just by fluctuations. This means that the sample handling has to be very good so that you don't lose any analyte. And also means that it's critical to have a way to qualify the samples, as I'll show you later on, at least in one case, because we have to make sure there is analyte in the sample. Otherwise, we just get false negatives. So what makes this problem worse is that different individuals have very different amounts of DNA in their blood, either normally or in pregnancy or disease. And we don't know why. So in pregnant women, commonly in the population, we see a 20-fold range in the amounts of fetal DNA. And in cancer patients, we see at least a 100-fold range in the amount of tumor-specific DNA in the blood. And the reasons for this remain mysterious, in spite of many attempts to decipher them. So to do reliable qualitative measurements, and to do any quantitative measurements on DNA molecules in blood, we have to overcome stochastic noise. And there are two ways to do this. The easiest one is to repeat the measurements. And if you repeat the measurements, then you can improve the signal to noise by roughly the square root of the number of measurements you make. So if you repeat 100 measurements, then you can improve the signal to noise by a factor of 10. But our DNA measurements consume the sample. So if I require a 10 ml blood sample for a single measurement, then for 100 measurements, I require a liter of blood. And that's impossible. Okay? So I can't do that, not in the real world. My alternative is to try to combine results from different but biologically equivalent markers. So for example, if I'm looking at a chromosomal aneuploidy, any sequences that come from the same chromosome are equivalent because they allow me in the same way to count chromosomes. And in a much more sophisticated way, I can use haplotype information to try to combine measurements from a region of a gene rather than have to make a direct single gene measurement, and I can improve my signal to noise that way substantially. So very briefly, let me show you the status of 3 potential areas where non-invasive measurements of DNA in plasma appear to be useful. So in prenatal diagnostics, this is a commercially available product from several companies in the United States, including Sequenome. We can test for common aneuploidies, as you'll see. We can test for rhesus D incompatibility. And in the future, this approach is going to expand to microdeletion syndromes and to clinically actionable single gene disorders, to Mendelian disorders. In fact, it's possible to reconstruct the entire genome sequence of an unborn fetus, but this is still too expensive to be used in routine practice. But as sequencing cost drops, it's going to become feasible. And these methods use generally known or inferred haplotype information. They average results in regions of the genome. So this is a mature commercial product. Non-invasive cancer testing is a research tool, but it looks extremely promising. I predict within a few years it will become common in clinical practice. The current focus is on somatic mutations that make tumors sensitive to resistant to drugs, and I'll show you some examples of this. What's usually done in this case is to start with a patient biopsy of the tumor and characterize in detail sequence abnormalities of that individual patient's tumor, and then choose glaring abnormalities to study in more detail in successive measurements from blood. And once again, for detection, individual markers can suffice in those cases where cancer is abundant. Cancer DNA in the blood is abundant. But otherwise, we need to aggregate results from multiple markers. Other diseases are just a dream at the moment, but in neurodegenerative disease, there's massive cell death in the brain. In autoimmune disease, inflammatory disease, cardiovascular disease, transplant rejection, all of this seems feasible. It's just that in these cases, all of them, there probably are no sequence differences in DNA that comes from the target tissue. So one will have to use epigenetics instead of sequence, but it's feasible. Okay, so the focus of the rest of this talk is not on methodology, but on results. But just before I get there, how do you do these measurements? Well, it depends on the complexity of the measurement. If you need massive amounts of genome sequence information, you have to do contemporary genome sequencing. If you need information on tens or hundreds of loci, but not more, DNA mass spectrometry, which I'll show you a bit of in this talk, is useful. And if you're just interested in a single locus, then PCR is fine. The relative considerations among these 3 methods are your need to quantify or not, error rates, cost, turnaround time. And the major disadvantage of DNA sequencing at present seems to be the enormous amount of data processing that's needed to handle results of a typical clinical study. Okay, so we transition now to non-invasive prenatal tests. As I mentioned, and as you'll see, this is a mature commercial product in widespread use in the United States. The procedure is to draw 1 or more 10 cc samples of maternal blood. From this, at Sequenome, we isolate 4 cc's of plasma. And we're going to use random shotgun sequencing to characterize the DNA fragments in blood and compare the sequence of these fragments with a genome assembly. Very straightforward, but a lot of data. Now, an average pregnancy, it turns out, is 13% fetal DNA and 87% from the mother. But as I've mentioned already, there's almost a 20-fold range in the fraction of DNA that's fetally derived. And so we must have a control to ensure that there's sufficient fetal DNA to detect traits of interest. We have to qualify the sample, as I've mentioned. Otherwise, we may erroneously conclude that the fetus is female because the mother's female, and that the fetus is normal because the mother's normal. In other words, we have a significant risk of false negatives. Now, for male fetuses, we can use Y chromosomal sequences because the mother's female. She doesn't have a Y chromosome. But half of all fetuses are female, and we can't use that trick. And so, in fact, the actual method that we rely on currently is epigenetic signatures. We look for DNA sequences that are methylated in the fetus, but not in the mother. These are imprinted sequences whose methylation is necessary for proper fetal development. So there are a variety of methods that one could use, but this shows what we actually do. We search in the genome for sequences that are methylated in the fetus, unmethylated in the mother, and that fall in the recognition pattern of restriction enzymes that are methylation sensitive. So that when we then take the mixture of maternal and fetal DNA from a pregnant woman and digest through the restriction enzymes, we can destroy the maternal DNA, shown here in the open circles, and just preserve the fetal DNA in the dark circles. And when we quantify that by a competitive PCR and detect the products by mass spectrometry, we have a very good way of estimating the amount of fetal DNA. Now, in practice, we do this for several different methylated regions with a bunch of internal controls. But I'm going to show you just what one of these imprinted regions looks like to convince you that this is a very clean and powerful method of qualifying the samples. So this is one methylated sequence that, as you can see in women who are not pregnant, there's no methylated DNA. In pregnant women, there's a significant signal from methylated DNA. We can detect even before 8 weeks, and beyond 8 weeks, it's a fairly robust signal. But I want to point out two things. Enormous variability from individual to individual. And look at the scale. This is the number of molecules of that sequence in our plasma sample. And it's 140 molecules. Okay? So we are in the domain of stochastic noise. And we need to use the methods I described, at least in principle, to try to compensate for some of that stochastic noise to get good quantitative accuracy. So I'll focus a bit on trisomy 21 because it's the most common fetal aneuploidy, but the methods are general to any kind of copy number abnormality in a fetus. Here's a cartoon of a sample of the mother's blood, and I've colored the fetal sequences red and the maternal sequences blue. But in reality, with current single-pass sequencing on these fragments, we don't have enough accuracy to tell the difference between the mother and the fetus. All we can do is look at the sequence and try to assign it to a chromosome. So this cartoon shows the experiment that we do in routine non-invasive prenatal diagnostics from the mother's blood. We sequence fragments from one end randomly. We sequence 10 to 15 million such fragments, and we use these to build up the relative distribution of, let's try this again. I'm afraid we have to let this click through. I have no way to speed it up here. We're going to build up the relative distribution of fragments from each chromosome in the mother's blood. And your intuition should correctly tell you that if the fragments come from a large chromosome, there'll be relatively many of them. And if the fragments come from a small chromosome, there'll be relatively few. As some of these fragments come from the fetus and some come from the mother, but we can't tell the difference. So in the end, we have a plot of the relative abundance of fragments from each chromosome, and that's all we get out of this measurement. If we focus on a single chromosome, if the fetus is normal, then we expect to see a certain number of fragments proportional to the size of that chromosome. But if the fetus is abnormal, then there'll be extra fragments, more than we expect to see, because the fetus will contribute too much. The mother's normal, presumably, so she does not contribute too much, but the fetus does. Okay, so to prove that this test was useful with Women's and Infants Hospital in Rhode Island, we performed a clinical trial of about 2,000 patients at high risk for chromosomal aneuploidies. And this shows the results of that trial for the three common trisomies, trisomy 21, trisomy 13, and trisomy 18. In each case, what I'm plotting is a measure of the statistical normality or deviations from normality of the representation of fragments of those chromosomes as a fraction of the independently measured percentage of DNA fragments in the mother's blood that come from the fetus. And as expected, the pregnancies in which there's a lot of fetal DNA have very robust signals, and the fragments from pregnancies in which there's very little fetal DNA, they have very weak signals. We effectively use a cutoff of 4% fetal DNA. If a woman has less than that, we won't do an analysis. And that means that we have to reject about 1% of the samples, but it means we can get definitive results on 99%. And you can see by and large that there's an excellent discrimination in each case between the roughly Gaussian distribution of normal fetuses and the overrepresented samples from aneuploid. So when this data was analyzed blindly, the performance was extremely good. The tests are not perfect, but they're very close to perfect with specificities and sensitivities usually in excess of 99%. That original clinical trial looked at the three common trisomies. It's been extended to twins and triplets to do gender determination and to do the common XY aneuploidies. And the current commercial product has available all this information. Whether parents wish to get gender information is, of course, optional. So this shows, as of 3 or 4 months ago, the status of our commercial clinical test, which was independently developed by our CLIA lab based on the results of those clinical trials. This is roughly 100,000 patients. We're roughly testing patients at the rate of about 150,000 a year now. And the results are really tremendously good. But it's important to understand that even though this is far and away more accurate than almost any other clinical diagnostic test commercially available, it is not perfect. We will make occasional mistakes, false negatives, in patients that have very low percentages of fetal DNA. That's the extremes of stochastic noise, and there's nothing we can do about that. We will also have occasional false positives, and those are interesting. They usually speak to biological peculiarities in the sample. So there's a phenomenon called confined placental mosaicism, in which the placenta, which is the source of most of the fetal DNA that we see, is aneuploid, but the fetus is normal. We'll always get those wrong. If you start out with two twins, one of which is aneuploid and one of which is normal, and you look too early in pregnancy after one has died, the placenta will still represent the twin that's no longer there, at least in part, and you'll have a false positive. And we can also have other more complicated issues. But these errors aside, we do very well. The one complication I want to focus on, which shouldn't have been a surprise to us, but may have been, is what happens if the mother, in addition to being pregnant, has cancer. Well, then, on top of the fetal contribution in her blood, there will be a tumor contribution in her blood. And let me show you what one of these patients looks like. A large amount of apparent fetal fraction, because we didn't know there was anything else there. An indication of trisomy 21, but negative z-scores mean chromosome deletion, an indication of loss of chromosome 13 and chromosome 18. A fetus with these characteristics couldn't survive. And it turns out that postpartum, this patient was diagnosed with metastatic carcinoma. Here's another case. We actually didn't believe it, so we repeated it. It gives you some idea of the results here. Again, this mother apparently had a fetus that was lacking parts of chromosome 21 and 13 and had too much 18, but in reality, this mother had invasive angiosarcoma. Okay, so you begin to see that when we use a discovery tool like sequencing in a clinical setting, we discover not only what we were looking for, but we discover things that we weren't necessarily looking for. Here's such a patient. This shows the relative abundance of sequence across part of the genome from chromosome 7 to the X chromosome. And you can see that in this sample of plasma from a pregnant woman, there's too much of the short arm of chromosome 18. Now, when we see a case like this, and we see these often, we don't know whether it's an abnormality in the fetus, the mother, or both, unless it's a well-characterized clinical phenotype. Now, we can distinguish between the mother and the fetus by going back and looking at the mother's Buffy code, which is pure maternal. But these are a problem. Here's another 3 cases in which we see too much or too little of small regions of chromosomes, and we may not be able to tell whether this has a clinical implication or not, depending on how much is known before. So this is the future. As these tests evolve, you're going to be getting more and more detailed chromosomal information, and it's important that medicine keep up with our ability to predict the phenotype that results from these situations. Now, as I already mentioned, we published 2.5 years ago that it was technically feasible to work out the complete genome sequence of an unborn fetus from the mother's blood. That experiment was quite expensive. In the past year, 2 other groups repeated it, made some modifications and improvements. But this is far from being routine practice. And that's fortunate, because the implications, the ethical and social implications of being able to know the entire genome sequence of a fetus before birth are profound. And I think we need some time as a society to figure out how best to use this information before we get flooded with it. Okay, so that's what I wanted to tell you about non-invasive prenatal testing. And in the remainder of this talk, I'm going to show you the much less mature field of looking at tumor. And I'm going to focus here on using mass spectrometry, although there's a place in these measurements for sequencing and for PCR as well. But in most tumors, there are a relatively small number of somatic mutations that act as driver mutations and determine the properties of the tumor, and in many cases determine the drug sensitivity or lack thereof of the tumor. And the number of these falls into a range where in most cases, we don't need to do whole genome sequencing, but it's too complicated for PCR. And so DNA mass spectrometry plays a powerful role here. And what it basically allows us to do is a highly multiplexed PCR and then simultaneous detection of many, many informative PCR fragments. Because unlike optical or fluorescent detection, which is limited to several analytes in the same tube, mass spectrometry, because it's a high resolution spectrum, not a low resolution spectrum, allows potentially hundreds of analytes to be analyzed instead of just a few. Well, I want to show you just a couple of examples. In the past year, there have been many publications demonstrating the promise of plasma DNA measurements in following the course of cancer. The first of these that I became aware of came from Johan de Bono's group in London. And they're doing phase 1 clinical testing. So these are patients with quite advanced cancer. And they were characterizing cancer-specific DNA sequences in plasma and also comparing these with tumor biopsies. So this slide shows you the typical amount of DNA, on average, that we measure in pregnant women, here expressed in terms of nanograms of total DNA per mil, and that de Bono's group measured in healthy volunteers and late-stage cancer patients. The methods used were different, so I don't think that this difference between 3.8 nanograms and 7.4 is necessarily significant. But the methods used between these second and third sample were the same. And so the roughly 8-fold increase in the amount of DNA in late-stage cancer is tremendously significant. And it encourages one to suppose that most of this extra DNA is from the tumor itself, although that remains to be proven. So this shows the amount of DNA in various patients as a function of the type of cancer, colorectal, breast, melanoma, and so on, compared with the healthy volunteers. And you can see that as in pregnancy, this is a log scale, as in pregnancy, there's a tremendous inter-patient variation in the amounts of this DNA. And as I've already mentioned with pregnancy, the bottom, roughly half of these samples are in a domain where they're compromised. The results are compromised by stochastic noise. The rest are not. Now, de Bono had at the time available methods for detecting mutations in blood, or tumor, that were about 5 or 10% sensitive. Okay, so smaller fractional amounts of mutant sequences he couldn't reliably see. And this very complex slide shows in many different types of cancer and many different oncogene mutations the relative concordance of measurements from plasma and measurements from a sample of the solid tumor. And I can tell you that the concordance on average is roughly half. And that's probably because of two reasons. The first is that the average amounts of these mutant sequences are low, and probably the samples where there's a small amount of mutation are missed either in the tumor or in the plasma. Okay, so that's one reason for discordance. But the other reason for discordance is that there's no reason to think that the DNA that derives from a tumor measured directly from a sample of the tumor will be the same as the DNA that derives from blood, because in blood you're looking at the cancer cells that have died, and in the tumor sample primarily you're looking at cancer cells that are alive. Okay, so we have a ways to go, and we're going to need more sensitive methods, which I'll show you briefly, to try to improve on this. But it turns out, curiously enough, that just the total amount of DNA in the patient's blood is a very good signature of prognosis in these cancer patients. And here, patients who have relatively small amounts of DNA in their blood seem to have a pretty good prognosis over a 2-year period, whereas patients who had the highest amounts of tumor DNA in their blood had a very poor prognosis. And this was one particular study, but these results have been seen now in other studies, okay? So this is good, but what we really want to do is be able to use these measurements to help guide therapy or to help better predict prognosis for individual patients. And so the second example I want to show you was recently published from a Japanese group using DNA mass spectrometry to look at a famous mutation, the T790M mutation in EGFR in lung cancer patients. So in lung cancer, mutations in EGFR make you a candidate for certain drugs that inhibit this tyrosine kinase. But patients who also have this additional mutation, T790M, are less susceptible to the drug. So the drug is less effective in these patients. So this is very useful, and the group measured it on about 100 patients. And what they found was in plasma, non-invasively, they could see this T790M mutation in a significant fraction of the patients. And those patients who were T790M positive, as shown in the red curves, had relatively poor prognosis compared with the patients who were T790M negative. So this is perhaps the first example, but example of many more to come, in which measurements of somatic mutations non-invasively in plasma are going to allow us to monitor cancer therapy and make choices to keep or switch drugs depending on the mutations that show up. But to make these measurements, we need higher sensitivity. And in fact, these measurements from Japan used a trick which raised the sensitivity to better than 1%. And I'm going to show you a new version of that trick just to let you peek a little bit into the future. This is a tool that we recently developed at Sequenome called ultrasensitive detection, and it's based on a very simple principle. We have a huge excess of wild-type sequence. We don't want to see it. We have a very small amount of mutant sequence, let's say 1%. That's what we want to see. This gives us a tremendous background in the mass spec. So since we're actually detecting sequencing by synthesis products after PCR, what we do is we leave out the substrate that can copy the wild-type template. So we have substrate that only copies the mutant template. That substrate has a biotin on it, and so we can use tryptophan to pull out the molecules that correspond to the mutant sequence. The wild-type sequence becomes invisible, and when we look, in this case by mass spectrometry, we see only the mutant sequence. So here's an example of how well that works in synthetic experiments for a bunch of different mutant sequences and a process control to prove there's DNA present. You can see that at 1%, 0.5%, even 0.25%, we get good strong signals. 0%, we get no false positives, and our process control shows there's DNA there. So these were just synthetic spike-in samples, but the clinical samples have now been studied by the same methods. And this tool looks very promising and should be available commercially in a few months. Now, at these very low levels of DNA sequence, detection is possible, but quantification is not because of stochastic knowledge. Now, in the last few minutes of this talk, I want to show you an example of where quantification, how much mutant sequence is present, is tremendously useful. But I can't do that on plasma. I have to actually go to a sample of the tumor to have enough material to work with. So I'm going to talk for the next minute or two about telling whether a tumor is homogeneous or heterogeneous. That is, whether it's clonal or not. So if I have a sample of tumor, it's a mixture of normal and tumor cells. And if all the tumor cells are identical, then whatever mutations I observe, I should observe in all of them. which means that the allele frequency of any mutation should be the same, because it's just the percent of tumor cells in the sample. But if I have a heterogeneous tumor, where different tumor cells have different mutations, then this simple result will not apply. So I'm going to show you some examples of this in lung cancer samples from a Japanese group. These are some famous lung cancer cell lines, just to warm up to this. So here's PC9 cells, which have two mutations, but you can see that their allele frequency is different. All of them have this one, but only half of them have that one. H1975, another lung cancer cell line, again, 3 different mutation frequencies, a derivative of PC9, PC9ZD. It's a mess, okay? So most of these commonly used lung cancer cell lines are not clonal. And this shouldn't surprise you, because the way we grow mammalian tissue culture samples in the laboratory is not by plating from single cells, but by just dilution, so that there's no reason why these cell lines should be clonal. They just represent the most fit mixture. And the same thing happens in some, but not all tumors. So these are lung cancer patients. If you look at patient 88 at the bottom, 2 different mutations, exactly the same allele frequency. Patient 81, the same story. But at the top, patient 25, most of the tumor has an EGFR mutation, but the KRAS and TP53 mutations are rare. So very different results in different patients. More patients, the same idea. At the bottom, patient 471, the 2 mutations are at the same allele frequency, but the next patient up, 407, very different. Okay. Now the reason why this is so important is that if you have a patient with a clonal tumor, it's possible that a monotherapy might work. But if you have a patient with a mixture of different types of tumor cells, then it's clear that an effective therapy is likely to have to be a combination therapy. And this is a very important lesson in managing the patients. But as I said, right now, the frustrating thing is that we're not able to do this from plasma. This requires an actual sample of the tumor. Okay, so very briefly, one last case. Instead of looking at sequence differences, we can look at methylation differences. This Swiss group, published these results 2 years ago for breast cancer. Again, using mass spectrometry. Here's a case where in the normal tissue, a particular sequence, this is in one of the BRCA genes, is unmethylated. As you can see on the right, very little methylation exists. In a sample of the pure tumor in the center, it's almost fully methylated. And in the blood of this patient, you can see that it really looks like the tumor and not like the normal tissue. This tells you 2 things: that there is a tumor, and that most of the DNA in the blood comes from the tumor. So the advantage of methylation is we can derive many, many methylation markers and use them in combination to try to overcome stochastic noise. In this particular study, 8 different markers were used. I won't dwell on the details, but using principal component analysis, they're able to distinguish in blood with pretty good reliability the patients who are normal from the patients who have blood cancer. Again, these are non-invasive methylation measurements made from blood. So this gives you a peek into the future. I like methylation very much because we're likely to have such a large number of markers. But admittedly, the tools available today to study methylation are a little bit more taxing than just to do sequencing. So I've given you an overview of the current state of the art of non-invasive testing in pregnancy, in cancer, and a hint that these same methods should be applicable in a variety of other diseases. That's the end of my talk, and I'd be delighted to try to deal with some of your questions. Let me see what we have here. Okay, so the first question is, why once in a life? What about mutations caused by environmental changes? Okay, that I guess I'm supposed to write the answer and also say it. Well, environmentally caused mutations are rare. And just like epigenetics, if they lead to a disease phenotype as a result of occurring in a specific tissue, we have to access the specific tissue, and that can be very difficult. So I'm not saying this is not useful, but I'm saying this is a painful way to make a living. So the second question is, do we collect tissue samples or blood samples? And in pregnancy, blood samples only. In cancer, it's nice to have both tumor and blood. Okay, which DNA purification do we use from whole blood? Okay, let me answer that. We first make plasma. Let me correct my spelling here. Okay, so that's straightforward. Okay, the next question is a very sophisticated one. Has to do with does the nature of the chromatin in the cells affect the efficiency of producing fragments? And the answer is it does affect it, and we're trying to work that out. Okay, the next one. Let me just. Okay, this is also a very, very good question. It has to do with how do we manage to get good enough signal to noise to detect microdeletions? And the answer is, and then I'll type it out. We have to sequence much deeper to see these microdeletions, and we have to use we have to use fancy signal processing methods to get these answers properly. Okay, I think this one we don't need to answer because it looks like it's already handled. Okay, this is a question about mitochondrial DNA, if I understand it right, and let me answer that. We have no straightforward way to look at fetally derived mitochondrial DNA because it's the same as it comes from the mother, so it's the same sequence of the mother. This is about the intellectual property in this area. It's a big contest right now. Okay, I think I've answered. Oh, I see. Answer the questions verbally, not typed. Oh my goodness. Okay, so okay, let me go back over some of these. A question, do I think direct DNA or RNA purification from whole blood is desired? And my answer is that it's much easier to work with plasma because when you work directly with blood, it's you have too much of a danger of breaking the host cells. Going backwards, the question was, do I think a single or a few genes could account for different tumor metastatic potential? I really haven't talked about metastasis at all, and I have no idea how complicated that is. Let's see if I can. I guess I can't get back to any of the others. Okay, I think we've answered all the questions, and I thank you, anybody still listening, for participating in this. And this talk can provide CME credits. So those of you who want CME credits should go to this website and follow the instructions once you log in to get your CME credits. I guess that's the end. Let me just check and see if there's any more questions. Oh, here it goes. For IP, have I mentioned the answer? The IP situation in the non-invasive prenatal space is extremely complicated, and there's a great deal of litigation. In the cancer space, the IP situation is also complicated, and there's no commercial product yet, so there isn't a great deal of litigation, but there probably will be in the future. Okay, I think I'm done, and all I need to do is to figure out how to log off here.